Hello guys, today my topic of discussion is Lac Operan Polycistronic messenger RNA. So I will explain you about the Lac Operan and the polycistronic messenger RNA. So what is Lac Operan? So Lac Operan is basically a machinery, you can say, the E. coli utilized to produce energy from the lactose when glucose is not available. So Lac Operan is basically a machinery, you can say, which is present in the genes, in the chromosomes and the DNA of the E. coli, and E. coli get energy when it start this Lac Operan. And what are the conditions when he don't have the glucose and when the lactose is available, right? So let me get my pointer. So here is your E. coli, here is the plasmid of the E. coli. So this is glucose. So when there is a high glucose, right, high glucose available, so this glucose E. coli utilize to get energy, right? So whenever the E. coli have high glucose, it causes inhibition of this adenyl cyclase. So down the road, whatever happen is knockout. That is one point. So when number one, when the E. coli have high glucose, it causes inhibition of the adenyl cyclase, and this cap protein activation, all this process will be knocked out. Second thing, when there is a high glucose, the LAC-L gene will be synthesized repressor protein, right? LAC-L genes will synthesize the repressor protein, and if this repressor protein go and bind to the operator site, so there will be no activation of this Lac operon, right? So this is two things happen when the E. coli is having high glucose. So high glucose, adenyl cyclase inhibition, one thing, no cap protein activation. The second thing is LAC will synthesize the repressor protein, and these repressor protein go to the operator side bind. So this whole Lac operon will be shut down. So that is happening when there is a high glucose, right? So now what is happen when there is a low glucose, right? So now you have to understand what is happening when there is a low glucose. Let me get my pointer. So here you go. So now you have to write down over here low glucose in your book. So when there is a low glucose, right, for the E. coli need to get energy. So what it will do, it causes activation of this adenyl cyclase. So when there is a low glucose available for the E. coli, so E. coli will does what? It causes activation of the adenyl cyclase. So what is happening? ATP is converted into cyclic AMP. Cyclic AMP causes the activation of this CAP protein. What is CAP protein? Catabolic activator protein. It causes this catabolic activator protein to bind to the CAP site. So here it comes to the bind to this site and causes activation or induction of the transcription. So what is transcription? This is your this is the RNA polymerase of the E. coli. It start producing the genes or start making the genes, and these genes will be required to form the enzymes, and then these enzymes will be utilized for the E. coli, whatever the purpose. So what you have to remember, low glucose causes activation of the acyl cyclase and activation of the CAP protein, and CAP go to the CAP site. So low glucose, you have to remember CAP activation, right? So second thing is when low glucose and lactose is available, right? Lactose is available. So when lactose is available, what would be happen? So one thing when low glucose, so what is happening? The transcription cap protein bind over here and activate the transcription. Now repressor protein is on the operator. You need to remove this protein so that transcription will be go on. So what will be happen when lactose is available? This allolactose, it remove this protein, it bind with this one. This is your allolactose. Now your what will happen? This operator site is now open. No repressor protein is here because it causes inactivation of the repressor protein. Now what is happening? CAP causes activation of RNA polymerase, and RNA then causes synthesis of the enzyme by these genes. So LAC-Z, it produces beta galactosidase, LAC-Y permease LAC-A. It causes the synthesis of the beta galactosidase transacetylase. I will let you know about the function of these genes. So what you have to need to remember, when there is the high glucose, adenyl cyclase is inhibited, right? And what the second thing, repressor protein go and bind to the operator. That is happening when there is a high glucose, right? So when there is a low glucose, what will be happen? Adenyl cyclase get activated, ATP converted cyclic AMP, cap will be activated, cap bind to the cap site, and what will be happen? Lactose causes inactivation of the repressor protein by binding with the repressor protein. So this is how the expression of the genes will be happen. So this machinery, the E. coli is using when there is a low glucose, because E. coli need the energy. So the energy E. coli getting is from the lactose, and lactose is your disaccharide. Don't forget about that one, right? So this is what happens about the situations. So what I told you, when you have low glucose, when the bacteria have low glucose and lactose available. So low glucose, you have to remember cap will bind. One thing, whenever low glucose, you see this one structure, it will be bind. When there is a lactose available, as I told you, when there is a lactose, it go and bind with the repressor protein, right? This one. So repressor will be out. So that's why on the operator side, you will not see the repressor protein. It will be knocked out because lactose go and bind with this one. So that is one thing you have to remember when there is a low glucose and lactose available. So in this condition, when both are cap is binding with the cap site and repressor protein is removed, lag genes are strongly expressed. So they will make the enzyme, and those enzymes do their work which is needed by the E. coli to get energy. Now that is one condition. Now you can see when high glucose. So what would be happen? Correct. So when there is a high glucose, this cap will be not activated because high glucose causes inhibition of the adenyl cyclase enzyme, right? So cap will be not activated. You see over here, cap will be not available. Now lactose unavailable. As you know that whenever the lactose available, it go and bind with this repressor protein and remove it from the operator side. So now you have to see these things and see what will be the action over here. High glucose, no cap will be available. Lactose unavailable, repressor will stay bind because it is not removed. Lactose is necessary to remove this repressor protein, as it is bind over here and remove it. So what will be the expression? Lag genes not expressed because this is repressor protein bind with the operator, not let the genes to be expressed. Now you see the low glucose. As I told you, whenever the low glucose, this cap will be present. So cap is here, you go. Now low glucose, adenyl cyclic activation, cap will bind to the cap side. Now lactose unavailable. Again, whenever lactose is unavailable, repressors stay on the operator side. So if low glucose, cap is binding. That is the one point. But lactose is unavailable because lactose is necessary to bind with this repressor protein and remove it from the operator side. When it is removed, then cap can do the work and causes the gene activation and all that process. So what will be happen? Lag genes will be not expressed because of this repressor. And why repressors stay on the operator side? Because lactose is unavailable. Now the third condition, high glucose. Fourth condition, sorry. So whenever high glucose, CAV will be not activated. As you know that the high glucose causes inactivation of the adenyl cyclase, right? So it is not bind to the site. But lactose available. So whenever the lactose is available, you will not see the repressor protein on the operator side. Why? Because this lactose is go bind over here and remove the repressor protein from this side. So what will happen? Cap is there. It cause cap is not there, but glucose is there. Because why cap is not there? Because high glucose causes inhibition of adenyl cyclase and cap protein. Lactose available. Repressor is not on the side because all the lactose go and bind with the repressor protein and remove it from that side. So in this case, what is happening? Very low basal expression of the protein will be available. That is one point. So what they're saying over here, classic example of genetic response to the environmental change. What you have to remember, that when glucose is absent, when glucose will be absent, lactose is available, the Lac Operan activated to switch to the lactose metabolism. This is called mechanism of shift. So what has happened? Low glucose, adenyl cyclase, cyclic AMP, cap protein activation, transcription. High lactose unbinds suppressor protein from the repressor at the operator site, increased transcription. So they both will be necessary for the strong expression of the genes, right? So that is Lac Operan. What the next thing I want you to know, that is the Lac genes. So Lac Z synthesize beta galactosidase. So what is the function of this enzyme? Increase lactose to convert into glucose and the galactose. That is the function of this gene. Then the second is LAC-Y. So LAC-Y form the permease protein. permease enzyme. It causes increased cell permeability to the lactose, right? So when these genes will be expressed, they do formation of these proteins, and these are the function of those proteins which E. coli use when glucose is not available. Now the third one gene is lac Lac A. So Lac A is synthesizing beta-galactoside trans-acetylase, as the enzyme is indicating its function. Trans means transfer and acetylase means acetyl group transfer. So transfer the acetyl group to beta-galactosides, not necessary for the lactose metabolism by the E. coli. This is the job of this enzyme protein. Now the last point which I want you guys to know about, what is polycistronic messenger RNA? You what is asking this question, right? So in a eukaryote, what is happening? One messenger RNA codes for the two enzyme. But in prokaryote, like E. coli, one messenger RNA codes for three different enzymes. Three enzymes. So what are the three enzymes which I show you over here? Let me go back to the picture. This one. So these are the three enzymes. These three genes, one, two, three. These three genes express three different enzymes, right? So one messenger RNA code for the three different protein A, B, and C. So that is called the polycistronic messenger RNA. That is one E. coli, one messenger RNA code for enzyme. The genes as GYA. So E. coli messenger RNA is the example of polycistronic messenger RNA. Like one messenger RNA work to form many proteins or enzymes which carry out the E. coli function. Hope this make you understand well. Thank you so much. Stay tuned. Bye-bye.